Hello guys, this is Mukherjee, your biology educator, and today I'll discuss with you the C3 cycle of photosynthesis and higher plants, a topic which we study in class 11th NCERT biology. So before we dive into the entire process of the C3 cycle, what exactly is photosynthesis? From where I gave the introduction. Photosynthesis, if you break this word, photo plus synthesis means something is getting synthesized, something is getting produced or to make something in the presence of light. So what is photosynthesis? It is the formation of food in the presence of light by the green tissues of the plant. This is the definition of photosynthesis. But in broader terms, since you are grown up kids, now photosynthesis takes place in two events, that is the light reactions and the dark reactions. What happens in the light reaction? In the light reaction, the light is being captured by the green tissues of the plants, as I told you. That is the grana, which captures the light, where the light reactions takes place. That is the site of the light reaction, the grana. The site of the dark reaction, it is the stroma. So, during the light reactions, the light is being captured, and in the process, ATP and NADPH, ATP and NADPH are energy intermediates which are being utilized during the dark reaction to form the CO2 molecule is converted into the carbohydrate, basically to form the food. The carbohydrate or the glucose is basically, you can say that carbohydrate is basically glucose that is being formed in the process. And in this process of converting CO2 into carbohydrate, this ATP and NADPH that is formed in the light reaction that is being utilized to convert the CO2 into carbohydrate. That is why, since a biomolecule is being synthesized in this process, the dark reaction, it is also called as the biosynthetic phase. Biosynthetic phase, why? Because a biomolecule is formed, that is the glucose molecule is being formed, so it is called as the biosynthetic phase. Based on a scientist who did the initial studies of the dark reaction is also called as the Blackman's reaction. So Blackman was a scientist who did the initial studies of the dark reaction, and in his name, Blackman's reaction, dark reaction is also called as Blackman's reaction. So there are also other synonyms that is being given to the dark reaction, that is the CO2 assimilation. That is, the CO2 is getting assimilated into the carbohydrate molecule, CO2 reduction cycle, because the CO2, if you see the oxidation number, the CO2 molecule is getting reduced into carbohydrate. So it is also called CO2 reduction cycle. It is also called as CO2 fixation cycle. That is the CO2 which is being taken from the atmosphere, it is getting fixed into the carbohydrate molecule. That is why the CO2 fixation cycle, it is also called as that. So there are various synonyms that have been given to the dark reaction. Now we will see the dark reaction altogether in a broad manner. If you're talking about the dark reaction, so based on the first stable product that is formed in the dark reaction, there are two types of dark reaction mainly. The first is the C3 cycle and the second is the C4 cycle. In the C3 cycle, the first stable product that is formed is a 3-carbon compound. In case of the C4 cycle, the first stable product that is formed is a 4-carbon compound. There is another kind of C4 cycle which is called as CAM plants. If you know, the deserted area, they have the CAM plants. They do the photosynthesis during the night. There also the C4 cycle runs, but only the photosynthesis happens during the night. And in the C4 cycle, the photosynthesis takes place in the day. The first stable product is a 4-carbon compound in both the cycles. And if you see the C3 cycle, the first stable compound is a 3-carbon compound. So in this lecture, we will discuss the C3 cycle. So if you see the C3 cycle, there are 3 events that take place mainly in the C3 cycle. The 3 events in the C3 cycle mainly, we will also discuss it during the cycle when we discuss the cycle. But first, let us first discuss over here. The first is carboxylation. In carboxylation, the CO2 is getting fixed. The CO2 molecule that is being accepted from the atmosphere, it is being fixed. Who fixes it? There is a 5 carbon ketose sugar, that is RUBP. Okay, RUBP, ribulose bisphosphate. Ribulose 1,5-bisphosphate. This is the molecule. This is a 5 carbon ketose sugar which will accept the CO2 molecule and it will fix into a 3 carbon compound which we call as the 3-PGA, 3-phosphoglyceric acid. This is the first stable product that is formed in the C3 cycle. The second step is the reduction. The second step is the reduction. What is being reduced in this process? If you see in the cycle when we will discuss, there is a carboxylic acid that is formed during the cycle. That is being reduced into an aldehyde molecule. So that is the reduction phase. And the third will be regeneration. What is being regenerated? This RUBP molecule that was fixing the CO2 molecule, that is being regenerated in the cycle. Now we will discuss these three events in the cycle, that is C3 cycle. Now when we are studying about the C3 cycle, there is a name of a scientist. He was an exemplary scientist who discovered the C3 cycle, and based on his name, this C3 cycle is also named as the Kelvin cycle. Kelvin cycle was named after an exemplary scientist whose name was Melvin Kelvin. What he did, he used a radioisotopic carbon, that is C14, to find out that the first stable product that is being formed in the C3 cycle is a 3-phosphoglyceric acid in a 3 carbon compound. So it was a very huge contribution to the field of biology. So on his name, this cycle is also known as the Kelvin cycle, and based on the first stable product that is formed, which is a 3 carbon compound, the cycle is also called as a C3 cycle. Now if we see the cycle, the CO2 molecule that is being taken from the atmosphere, that has been accepted by RUBP. I just said you that it is a 5 carbon ketose sugar, right? So if we number the carbon molecules, this is a 5 carbon molecule, this is obviously 1 carbon molecule, right? And in this case, a 3-phosphoglyceric acid, this is a 3 carbon compound. This is a 3 carbon compound. So if you see 5 carbon and 1 carbon, it is forming 6 carbon. So basically, there are 2 molecules of 3 carbon compound that is being formed. So basically, 2 molecules of 3-phosphoglyceric acid is being formed in this process. And the enzyme that is being used here is Rubisco. It is a very interesting enzyme. Why? Because ribulose bisphosphate carboxylase oxygenase. This enzyme can act as a carboxygenase as well as oxygenase, which means it can bind to carbon dioxide as well as oxygen molecules. It normally binds to CO2 molecules in the C3 cycle, but when there is a high concentration of oxygen or there is a high temperature, it can also bind to the oxygen, and there will be a wasteful process that will start, which is known as photorespiration, which is another topic we will study someday later. So in this reaction, the 5 carbon RUBP, which is the CO2 fixer, is fixing the CO2 molecules into 3-phosphoglyceric acid. In the next step, the 3-phosphoglyceric acid, it will utilize 2 ATP molecules. So 2 ATP molecules are, if I should write it in red so that you can see, the 2 ATP molecules are utilized in this reaction to form 1,3-bisphosphoglyceric acid. Again, this is a 3 carbon compound. So 2 molecules of 3 carbon compound is a 6 carbon. In the next reaction, 1,3-bisphosphoglyceric acid will convert, that is triose phosphate, or you can also say as phosphoglyceraldehyde. This is also a 3 carbon compound, and 2 molecules of 3 carbon compound is formed. In this process, since this is a reduction, phosphoglyceric acid is a carboxylic acid and an aldehyde molecule is being formed. So this is reduction, and the entire C3 cycle is a redox reaction. So here something is getting reduced, something else has to be oxidized. So the two NADPH molecule will get oxidized into NADP plus. This is the reduction phase. Now in the next, what happens? One of the triose phosphate molecules will release out from this reaction. So now here we had 6 carbon, we lost 1 carbon, so we are left with a 5 carbon molecule, that is ribulose phosphate. Ribulose 5-phosphate, which means that phosphate is present in the 5th position of this carbon, of this molecule, ribose molecule. So we can say that it is also a 5 carbon compound. Now, the next step is the regeneration step, in which the ribulose 5-phosphate will it will regenerate the RUBP molecule. Again, for this reaction, one ATP molecule will be utilized. So this was the C3 cycle that we discussed right now. Now, the events that I was talking about in the C3 cycle, that is the RUBP molecule getting converted into, it is fixing the CO2 molecule and the 3-PGA, that is the first stable product of the C3 cycle being formed. This is the carboxylation where the CO2 molecule is getting fixed. So this is the carboxylation step. Next is 1,3-bisphosphoglyceric acid is getting reduced into triose phosphate or phosphoglyceraldehyde, that is an aldehyde molecule. So this is the reduction phase. And here the ribulose 5-phosphate that is being regenerated into ribulose 1,5-bisphosphate. This is the regeneration step. So these are the 3 events that I was talking about in the C3 cycle that takes place: carboxylation, reduction, and regeneration. Now we will see the energy dynamics or the energy sheet of the C3 cycle, which is very important from the exam point of view, that is basically asked in the NEET exams or any other competitive examination. So here we see in the carboxylation step, there are 2 ATP molecules that are being utilized, and in the regeneration step, one ATP molecule is being utilized. So we can say that in the C3 cycle, there are three ATP molecules that are being utilized and 2 NADPH. Here in the reduction phase, there are 2 NADPH molecules that are being utilized. So when we start with one CO2 molecule, there are 3 ATP and 2 NADPH that is required. But if you want to form one glucose molecule, which is a 6 carbon compound, you will have to run the cycle 6 times. So we will need 6 Kelvin cycles, right? So this is multiplied, that is the 3 ATP will be multiplied into 6 and 2 NADPH will be multiplied into 6 to form 18 ATP and 12 NADPH for 1 Kelvin cycle. For 6 Kelvin cycle, that is for the formation of one glucose molecule, we need 18 ATP and 12 NADPH. When we started with one CO2 molecule, we needed 3 ATP and 2 NADPH. So if you are asked for the formation of one glucose molecule, you will write 18 ATP and 12 NADPH. If you are asked about per Kelvin cycle, then you will say that there are 3 ATP and 2 NADPH that are being utilized. You are often being confused in the numbers, so always keep a check what is being asked in the question. So this was the entire C3 cycle and the energy dynamics. I hope you liked the video and understood. Thank you.